A warm welcome to all the dear learners who have come to my channel, Microbiology Tutorials. Today I am going to discuss about the topic called as the DNA microarray. DNA microarray is a new technology which have been started or came into the existence in the era of 1990s. Before proceeding to the lecture, I would like to address some of the key issues about this technology, which have become an important tool in diagnostics also. If the medical students would be knowing about the ELISA technique, they would have been aware that in ELISA, we uses antibodies which are bind to the microtiter plates. And then we use these microtiter plates which has been bound, in which antibodies are bound to detect any specific antigens or antibody in the serum or other body fluids. So, and it is based on the enzyme linked immunosorbent assay, enzyme substrate reaction, which gives, if positive, except in competitive ELISA, where no color formation is given as a positive reaction, whereas in other, either it is direct, indirect, or sandwich ELISA, all forms, you see that they give a colored reaction. Or we go for another immunoassay techniques in which either radioisotopes are used, or in the fluorescent microscopy, fluorophores or fluorescent through fluorescent microscopy, antigen and antibody are detected. So it is also an advanced technology where instead of enzyme immunoassays, similar to technology which I discussed already about the ELISA or radioimmunoassay or fluorescent microscopy, here basically we use glass plates or nylon plates, which is coated with with genes, some of the important genes which we have to detect. Now, as we know that the DNA, which is used in the conventional PCR techniques, are based on the complementary base pairs with oligonucleotide primers. Similar technology is used here also. So basically what we do, that any gene of interest which we want to find are first, they are they are first of all amplified in the PCR, through PCR, and then these are treated with these plates already having the complementary base pairs. And if they complement each other or base pairs are matching together, they bind and they give different color bands, and through which we easily detect the desired sequence. So one thing is important here, that first of all, we have to uh, amplify those genes of interest. One thing more I would like to add, that what is the importance of all these molecular diagnostic techniques? Now, as we know that after the completion of Human Genome Project, we came to know about the entire sequence of the human gene. And as per the central dogma of molecular biology, we know that the DNA when they replicate, they make another DNA, and from DNA, mRNA is synthesized through transcription, and from transcription, translation of these mRNA is done into proteins, which can be in different forms. We know that the proteins are the building blocks of the body, and it constitute major enzymes, hormones, factors, all those important proteins which regulate our body functions. So instead of quantifying or knowing about the amount of concentration of different proteins, if we detect it at the genetic level, any defect, it would be quite easier for us to understand the problem or any defect at the genetic level. So it becomes quite easier for us to go to the root cause of the problem, and also from the diagnostic and prognosis, it also becomes easier for a clinicians to diagnose and to diagnose all those conditions at a gene level. So we know that gene have all the information coded in the coded form, and if we decode if we study on the genetic level, it would become quite easier to decode some of the major disorders or defects in a human, in a host body. Next important thing about this molecular technology is that it becomes quite easier to sequence the bacterial or viral or any other microorganism genome, and we can easily identify or sequence those genomes, and we can use it for the study on the pathogenetic effect, pathogenesis, or the root of pathogenesis or mechanism of pathogenesis of these pathogens. And it also helps through this, we can target those gene on the products which are made by, obviously these are made by the genes, so we can directly target and alter those genes so that they cannot make such products, or we can say any enzymes or toxins which causes harm or pathogenesis in the humans. So these are the some of the various important factors which are related to the study of any type of gene study, genetic studies, and also their expressions. So continuing to our chapter, now we will see that similar to PCR or other technologies or genetic technologies, how this DNA microarray are used in today's advanced diagnostic techniques, and how it is helping in the medical science. So today's topic is on the DNA microarray. So what is a microarray? So microarray is a slide or membrane with numerous probes that represent various genes of some biological species. Probes are either oligonucleotides that range in length from 25 to 60 bases, or cDNA. cDNA are those DNA which are made through the process of reverse transcriptase. Means that first DNA, either they are in the form of RNA, or they are converted from DNA to mRNA, and from mRNA through the reverse transcriptase enzyme they are converted into cDNA. It is similar to the mechanism of the retroviruses like HIV, HTLV, which uses this technique for spreading themselves in the body. So These are basically the probes which are used on these, as I told you, they are identical to the antibodies used in the ELISA technique. So probes are either oligonucleotides that range in length from 25 to 60 bases, or cDNA clones with length from 100 to several thousand bases. The array type corresponds to a list of reference genes on the macroarray with annotations. For example, 22K Agilent oligo array or NIA 15K cDNA membrane array. These are some of the examples of the microarray. Many individual users want to add their own array types to the list. What happens to a microarray? A microarrays are hybridized with labeled cDNA synthesized from mRNA sample of some tissues. The intensity of label, radioactive or fluorescent, of each spot on a microarray indicates the expression of each gene. One die arrays, usually with radioactive level, show the absolute expression level of each gene. Two die arrays, fluorescent label only, can indicate relative expression level of the same gene in two samples that are labeled with different colors and mixed before hybridization. One of these samples can be universal reference, which helps to compare samples that were hybridized on different arrays. Now, how these arrays used to, first of all, as I already told you the mechanism, that basically there is a hybridization. It is based on the hybridization process, means that two DNA are attached to each other, and if they are complementary base pairs exist on them, so they will form a double-stranded DNA. And if they are not complementary, then they will not attach to each other and will not get hybridized. Then next is the that some labeled, they are usually labeled either with they are fluorescent label, or which is a two die array, or one die array, which is where they are radioactively labeled here. Now these are the important parts. You can say that it acts as an indicator to for us to easily visualize any type of hybridization reactions. Now, DNA microarray is one of the most recent and advanced molecular diagnostic tests for the comprehensive analysis of etiological agents of disease. Microarray diagnosis work through the principle of hybridization of nucleotides from patient sample to oligonucleotide probes. Microarrays are now used for pathogen genotyping and identification, diagnosis of cancer, detection of antibiotic resistance through by checking that which genes have modified themselves so that they are becoming in the microorganism, so they are resistant to the antibiotic, or detection of gene mutations and diagnosis of the recent pandemic COVID-19. Oh, and here we can also study about the different type of mutations occurring in the gene. Either it is a point mutation, mostly in the single nucleotide as polymorphisms, or SNPs. Now, microarrays following use to detect and identify different microorganisms by targeting the intergenic transcript spacers, which are also called as ITSs, 16S bacterial and 28S fungal ribosomal subunits and others. In addition to them, they are also, we can use it for the ribosomal subunits because they are the important factors. Because ribosomes are the part where this transcription, translation process usually takes place. From the transcription to translation, these ribosomes play an important role. The advantage of microarray techniques is it have a high sensitivity and specificity and able to detect polymicrobial pathogens from a single specimen simultaneously, but it can only be used to identify to detect pathogens which are previously sequenced and have designed probes. Those novel species without previous known genome will not be detected. Nowadays, many DNA microarray techniques are used for the diagnosis of infectious diseases. Using this technology, the presence of one genome or cDNA sequence in 1 lakh or more sequence can be screened in single test. Now, next is here, you can see that usually this is also called as the DNA microarray as the gene chips or biochips. And why it is called? Because it's a collection of microscopic DNA spots attached to a solid surface. DNA microarrays are used to measure the expression levels of large number of genes simultaneously or to genotype multiple regions of gene. Each DNA spot contains picomoles, 10 to 12 moles of a specific DNA sequence known as probes or reporters. A probe can be a short section of a gene or other sample that are called target under high stringency conditions. The probe target hybridization event is usually detected in a quantifiable signal generation from the fluorophore, silver or chemiluminescence labeled targets to determine relative nucleic acid sequence in the target. Since an array can contain tens of thousands of probes, a microarray experiment can accomplish many genetic tests in parallel. Therefore, arrays have a dramatically accelerated many types of investigations in genomics. The major advantage is that here there are numerous probes are placed or they are attached to a solid surface and multiple genes, as you can see here, we can study through probe target hybridization process. And in one time, we can come to know about different type of genetic defects or mutations. Now, in standard microarrays, the probes are synthesized and then attached to a solid surface by covalent bond to a chemical matrix such as epoxysilane, aminosilane, lysine, polyacrylamide, or others. The solid surface in general is a glass or a silicon chip, in which case they are collectively known as Fe chip. When an Fe matrix chip is used, other microarray platforms such as Illumina use microscopic beads instead of the large solid support. Alternatively, microarrays can be constructed by the direct synthesis of oligonucleotide probes on solid probes surfaces. DNA arrays are different from other types of microarray only in that they either measure DNA or use DNA as a part of its detection system. So here you can easily see through the diagrammatic representation that how the DNA microarray mechanism works. So here is the substrate, then here linker through linker, and these are the DNA probes which are attached to the solid surface and how they are arranged sequentially on these surfaces. Now coming to the principal major technologies, what the technique here is used, that is either a cDNA probes is used, which are greater than 200 nucleotide, usually produced by PCR, attached to either nylon or glass supports, or oligonucleotides 25 to 80 nucleotides, then oligonucleotides 25 to 30 synthesized in situ on silica wafers. That is the Fe matrix, which we have already discussed, and then probes attached to tagged beads. Uses of microarray in DNA detections. DNA microarrays can be used to measure changes in expression levels, to detect single nucleotide polymorphism, or to genotype or resequence mutant genomes. Revolution in the analysis of genetic information. Hybridization is a highly parallel search by each molecule for matching partner on an affinity matrix. Specificity and affinity of complementary base pairing, use of glass as a substrate, fluorescence for detection, and the development of new technologies for synthesizing or depositing DNA have allowed the miniaturization of DNA arrays with increases in information content. DNA chip or DNA microarray allows to collect more information about DNA sequences in an afternoon than an army of scientists could collect in years using earlier several techniques. So as I already said that it is one of the most adopted, acceptable technique for where we can collect more information in a short period of time than any other test. So, or any other test where these information could quite take a longer duration. So DNA chips promise to carry the science of understanding genomes to a whole new level and to bring tools for getting DNA sequences to from the research labs to into doctors' clinics, and hence it can greatly help in the treatment and diagnosis of some of the major diseases, and help in also in identifying some of the major genetic disorders, major defects at the genetic level. Now here you can see through a diagram that what are the some of the important steps in the DNA microarray. So here you can see that there is the cells are obtained with genes that are needed for analysis. Then isolate the mRNA using extraction buffer, remove the buffer with RNA. The mRNA will be 5' to 3' with repeating AAA into. Convert each mRNA into colored cDNA targets labeled with fluorophores. With mRNA degraded, reverse cDNA is created, mixed colored samples of cDNA. Here you can see that how the mRNA is collected, and it is from the different forms. One is the conversion to fluorescently labeled cDNA, or you can convert it to fluorescent labeled cRNA, or hybridized, then convert to biotin labeled cRNA, or directly label the RNA with fluorescent tags. And here they can be used to hybridized to the array. Next is the what is the principle behind the DNA microarray. So nucleic acids are extracted from the clinical samples. Then, first of all, you should know that it is necessary that before proceeding to the DNA microarray, we have to amplify the DNA sequences or the genes which we need to identify. And obviously, these amplification can be only used by using the PCR techniques, using primers for possible pathogens which are able to cause the infection, and then labeled with a fluorescent dye and labeled nucleic acid are hybridized on a DNA microarray. Finally scanned to detect the sample's particular probe or DNA interactions by fluorescence. Direct fluorescence method or enzymatic detection can be used to identify the hybridization, producing a semi-qualitative result. Here, through one example, I can give you ideal example that in the case of the traditional drug resistant testing of Mycobacterium tuberculosis, clinical samples takes a long time. Usually because generation time of Mycobacterium tuberculosis is more than other bacteria. So first of all, the culture takes a quite longer period, and after that, if we test for their multi-drug resistance or any antibiotic resistance, it further prolong the detection time. So this can be overcome through the DNA microarray technique, and the best technique there is that it determine Mycobacterium tuberculosis antimicrobial resistance pattern by characterizing mutations in the RPOB and CATG genes that gives rifampin and isoniazide resistance. So if we directly study the gene of RPOB and CATG, which are responsible in which if any mutations takes place, then the major drugs of tuberculosis, that is rifampin and isoniazide, are resistant. So it can be easily identified. So microarray technology may also detect extended spectrum beta lactamase, ESBLs, and carbapinase, which are a major these days, which have become one of the important factors of MDR in Enterobacteriaceae or gram-negative bacteria. So through this technique, we can also study ESBL producing bacteria and carbapinamase producing Enterobacteriaceae and identification of resistance in Plasmodium falciparum due to single nucleotide polymorphism. For the detection of resistance genes in both gram-negative and gram-positive bacteria for different antibiotics, AMR direct flow chip kit DNA microarrays better and rapid assay. So these are some of the important examples which can be used for the detection of important resistance genes in the different pathogens towards the antibiotics. Now microarray DNA chip, every spot on the chip is represents a different coding sequence from different genes. Each spot on the chip is made of a DNA probe that can pair with the cDNA that was created. Incubation with the mixed cDNA and the chip DNA will yield some pairing. Wash off unbound cDNA to see what has bound to the microarray. Then visualizing bound cDNA, the slide with the microarray chip is placed inside a dark box where it is scanned with a high resolution laser that detects the bound fluorescent levels. The information and images are then sent to the computer for analysis. So this is the machine, and these are the spots which are called as the DNA microchip on a solid surface. So all these gives you can see here that it will create a ratio image, and each color gives expression of the different genes. So if any green color will come based on the kit which is used. Suppose if a red color is coming or green color is coming, it will show a greater affinity for the desired gene sequence. Or if red comes, then it will show less affinity. You can see here that different color bands is here shown, that here you can see that there are 4 copies of gene A and 1 copy of gene B. So here, if 1 copy is there, it is showing blue color. If 4 copies are there, so it will show a green color. So based on that, how many copies are present, we can easily identify through these gene spots. Now here is coming to the historical part or its discovery. So in the late 90s and 2000s, DNA array technology progressed rapidly as both new methods of production and fluorescent detection were adapted to the task. In addition, increases in our knowledge of the DNA sequences of multiple genomes provided the raw information necessary to assure that arrays could be made which fully represented the genes in a genome, all the sequence in a genome of a large fraction of the sequence variation in a genome. So this was this is, we can say, the one of the latest technology, and basically it was introduced in the late 90s or and 2000s, and it have made a great landmark in detecting some of the important defects or important genes responsible for several disorders, or we can say resistance in the resistance caused by the bacteria or other pathogens. And it is one of the latest emerging technology, or we can say one of the promising technology for the future. It can become a very good alternative to the conventional mode of treatment, and also it can give rapid results in comparison to the routine technologies or the test we are using here. Now here you can see these are some of the important chips, or some of the important solids gene color colored spots which are present on the solid surface. Now here is one such technology that either measures DNA or uses DNA as parts of its detection system. Now here I can say that it is based on the principle is based on nucleic acid hybridization, and complementary strands of DNA are joined together by hydrogen bonds to form a double stranded molecule. And the technique comprises of a DNA chip, preparation of a DNA chip, the experiment and collection and analysis of results. Collection of sample. A sample can, in this case, be defined as a cell or tissues of the organism. then mRNA, isolation of mRNA, then creation of labeled cDNA and hybridization, then collection analysis. The collection of data is done by using microarray scanner. This scanner consists of a laser, a computer, and a camera. The laser excites fluorescence of the cDNA generating signals. When the laser scans the array, the camera records the images produced. Then the computer stores the data and provides the results immediately. The data thus produced are then analyzed. The difference in the intensity of the colors for each spot determines the character of the gene in that particular spot. Types of DNA microarrays are cDNA microarrays, oligo-DNA microarrays, BSC microarrays, which uses template amplified by polymerase chain reaction as the probe, and SMP microarrays used to detect polymorphism within the populations. Application is to study transcriptomes and proteomes, diagnose pathogenic as well as genetic diseases in man, to identify microbes in the environment with the help of species-specific probe, to identify to genotype genomes through single nucleotide polymorphism analysis, to detect gene expression of mRNAs, then to measure changes in the level of gene expression to observe unique mutations. Now, they have also some limitations also, and some of the important points in this is that it takes a lot of time to analyze as the amount of data collected from each array will be huge. The results may be too complex to interpret as we see different color spots, so it becomes very little difficult to interpret them. The results are not always reproducible. This technology is too expensive. The array provides an indirect measure of relative concentration. So these are some of the limitations. Now the labeled ctRNA or cDNA are then hybridized. Already we have discussed this, and it's a very sensitive conventional high throughput tool to study the structure of complex microbial ecosystems and explore functional gene sets. Since Human Genome Project was completed in 2001, it paved a way toward forward functional genomics, and microarray is the most common tool used to study function of genes through parallel expression measurements of genome. And the microarray is carried out by complementary DNA microarray oligonucleotide microarray series serial analysis of gene expression SAGE. So SAGE is also an important type of microarray. Now DNA microarray uses microchips and with a collection of microscopic DNA spots and hybridization into two DNA strands, which are technology which is borrowed from southern blot procedures to simultaneously screen multiple regions of genome and measure the levels of expression. So we can say that is amalgamation of different technologies, and it is basically the different technologies are brought at uh, at one platform, and it has been tried to utilize all these technologies for accurate and faster treatment, and through and to uh, complexities, to remove any type of complexities or avoid any type of complexities included in any type of diagnostics. So with this, we conclude our today's lecture. I hope it would have been beneficial for you. And if you have liked my lecture, so please do like and subscribe my channel Microbiology Tutorials, and do continue watching my other videos also, which will keep me always motivated to bring new topics and new way methods of technologies or treatment methods used in today's world in the 21st century. So I hope it would have been an added advantage for you, or helped in your studies. Thank you. See you again.